But let's get started on proteins, right? So the first month we learned about cell biology, which was cells as a whole, right? How does life work? How does life exist? We learned about a little bit about DNA and how DNA, the genome keeps the cells alive, and how the DNA can define what a cell is and what type of cell a cell is. The thing is, DNA actually, in most cases, doesn't do shit, right? I'm sorry if there's any children watching this. I'll try to keep my swearing to a minimum. But DNA, it doesn't do anything, right? So when we think that DNA is this thing that defines what cells do, it's actually only a little bit true. There's also a lot of other stuff going on. And the most important thing that is going on is proteins, right? Proteins are what make everything alive. So like your ability to, flex your muscle like this, your ability to run around, the way your heart beats, the way E. coli cell does its thing, the way a cricket jumps, all of these things are due to proteins. Proteins are what make everything move, what make everything alive, what give everything the ability, every cell to do what it does. I'm not trying to overhype proteins, but proteins are amazing, right? Proteins are basically how you genetically modify things. Now, if you understand DNA, you can put DNA into cells, but if you can engineer proteins, you can do pretty much anything, right? Proteins can do almost anything you can imagine. The only thing proteins can't do that I know of are generate radioactivity, right? That's a pretty tough one. But they can generate light. Proteins can generate light. Proteins can absorb light. Proteins can fluoresce. Enzymes can break down almost every molecule. Enzymes can create almost any molecule. Proteins can transport almost any ion or molecule. Proteins can replicate DNA. Proteins can break apart DNA. Proteins can do basically anything you wanted, right? And that's the amazing thing about proteins. The only limit with proteins is our ability to understand how they work. Is there a particular reason why a protein couldn't generate radioactivity? There's not a particular reason. I guess if you could figure out a way to get, uh, well, because radioactivity it's a property of atomic nuclei. So I guess if you could get a protein to bind an atomic or radioactive nuclei, then you could generate radioactivity. Um, I don't, I'm not an expert on radioactivity, so I'm not going to be able to answer your question completely, Matthew. But I assume that's the reason. So like proteins are composed of just carbon, nitrogen, oxygen, hydrogen, sulfur, right? Now sometimes proteins bind other molecules, and sometimes proteins get phosphorylated, so they have phosphate atoms. Uh, but yeah, like so like hemoglobin, right? It binds heme, and heme has what's the atom at the center of the heme ring? Somebody help me out here. Manganese? I have no idea. It's been too long. Iron. That's right, iron. Yeah, so you can like have proteins that binds these other molecules, and then the protein, has these things, but generally proteins are only, composed of certain atoms. Now, proteins have 20 different amino acids generally that they are composed of. Now, you have to understand that in all biological cases, there's always exceptions, right? Biological systems are sloppy, they're messy, they're never exactly as you imagined them to be. So, the same is true in the case of proteins. So much so. And scientists have literally beaten this shit to death and trying to figure out like everything that goes on in proteins. Everything. And there's a lot of things that goes on. Not only are proteins composed of these amino acids, but sometimes proteins after they're made get what you call post-translationally modified. And what that means is they get like sugars added to them, they get other things added to them. Oh geesh, it gets super complicated. Super, super complicated. To where it can just be overwhelming and we don't understand what everything does, how much of an effect it has, how little of an effect it has, whatever. Um, there's even sometimes proteins get mistranslated. They get different wrong amino acids inserted into them uh, that can actually modify the protein in a certain way to respond to like immune events and other things. But we'll leave all that stuff alone. Now, the way proteins are made is they originally start out as DNA, right? So in DNA, what you have is you have a gene, and this gene tells the cell that it wants to make a protein. And it does that by having specific DNA nucleotides in a specific order, right? So it'll say, like, here's where I want the ribosome to bind to the DNA, and I want it to start making, uh, or not the ribosome binding to the DNA, ribosome binds to RNA. This is where I want um, the polymerase to bind to the DNA, and I want it to start making RNA, right? Because in order to make a protein, you have to make RNA first. What happens is the DNA is translated, they call it, into RNA. And then the RNA are transcribed into RNA, and the RNA is translated into protein, right? So DNA is transcribed into RNA. RNA is basically like DNA, except it has a uracil. That's the only difference. Well, no, the backbone has a 5 prime, I think it's 5 prime OH, but that's just complicated. We don't need to worry about that shit. What we need to worry about is just that DNA goes into RNA, goes into protein. Why are cells complicated like that? Oh geesh, I don't actually know. I don't actually know if anybody has a good reason why DNA goes into RNA, goes into protein, and why we just can't read directly from the DNA into protein. Um, if anybody has any good guesses or things they read, I'm all ears. Uh, I I'm sure people want to blame it on the RNA world and other stuff like that, or the fact that if like DNA gets mutated or damaged, um, then transcription helps prevent, I don't know, direct errors from DNA into protein or something. But we have a system set up. Yeah, Matthew. I think that's, you know, people say evolution always happens. DNA is more stable than RNA for sure. So RNA has this what they call, I think it's a 5 prime OH. It's not 3 prime. I'm pretty sure it's not 3 prime. It has it's more reactive. And so what happens with RNA is it's more likely to uh, autocatalytically cleave itself. Not to mention RNases are everywhere. So what happens is our bodies have developed self-defense mechanisms because a lot of viruses are made with RNA, right? So our bodies have developed these RNases, which are self-defense mechanisms that cleave RNA all the time. Um, yeah, Dave says cells have tend to have systems that preserve DNA, but it's more important that various RNAs that get degraded over time to control what proteins are synthesized and when. Yeah, RNAs do do allow a more finer tuning and control. Um, Finer tuning and control, not more stable. Esther, it should be less stable, right? Is there a 5' OH or something on the RNA? RNA should be less stable than DNA. DNA has an auto degradation, auto hydrolysis half life of like 50,000 years or something. DNA is really stable. Um, RNA is not as stable at all. But yeah, I like Dave's explanation. See, what happens is when you make transcribe DNA, you can transcribe it multiple times and have different copies of it. But I guess uh, you could regulate certain. I guess it gives you like another regulatory layer, maybe. RNA gives you a second regulatory layer above the DNA in which you can create other feedback loops. You can create um, load balancing, different stuff. But yeah, anyway, so when we get to the RNA, we get to the protein. Now, proteins are composed of amino acids, generally one of the 20 different amino acids um, that are part of life. Now, uh, when I was in my PhD, so DNA is like having a master branch where edits get made on RNA working versions, Chris. Um, kind of, kind of. Um, RNA is more like DNA is like I would say like the main process. And if you're thinking about programming, RNA are like the child processes. So it's like parallelizing, right? What you can do is you can make multiple copies of the DNA as RNA and make those into the cell, and then all those can be turned into protein at any time, kind of. But what you can do is you could kind of, you know, make as many proteins as you need, and then once you've made all the proteins you need, you could downregulate that RNA, um, make sure some of that RNA doesn't get expressed, gets degraded, gets silenced, or other things. So I think it's more like a way to respond to a quicker way to respond to maybe changes in cellular environment. But anyway, proteins. Um, there are 20 different amino acids, and they are all coded for by uh, well, not coded. Yeah, they're all coded for by three DNA bases. They call that a codon. So each amino acid, each three bases in DNA codes for a single amino acid, right? So if you have a 100 amino acid protein, that's going to be coded for by a 300 bases of DNA, right? There'll be like a stop codon at the end, so it'd actually be like 301 or 303 bases of DNA, but 300 bases of DNA codes for. Um, Shiv says, with many RNA from single DNA, what controls which protein it generates? So that's what the DNA codes in the RNA. So the RNA is just kind of like a copy of the DNA, right? Just think of it as a copy of the DNA that's not part of the chromosome. It's not permanent in the cell. Now, all the bases in the DNA, these triplet 3 base pair codons, these code for amino acids, right? Now, these amino acids, when they're strung together, they create proteins. You with me so far? Proteins, amino acids. Proteins are composed of strings of amino acids that are defined by the DNA, but that are actually translated from the RNA in the cell, right? So 20 different amino acids. If you want to be cool, you should memorize them all. Let me tell you. A is for alanine. There's no B amino acid. C is for cysteine. D is for aspartic acid. E is for glutamic acid. F is for phenylalanine. G is for glycine. H is for histidine. I is for isoleucine. There's no J. K is for lysine. L is for leucine. M is for methionine. N is for asparagine. There's no O. P is for proline. R is for arginine. S is for serine. T is for threonine. There's no U. V is for valine. W is for tryptophan. Y is for tyrosine. And there's no Z, right? Why is D for aspartic acid? Because A is for alanine. So you have to choose something else. Um, yeah, so yeah, Daniel, this is part of the central dogma, the standard dogma of molecular biology. DNA to RNA to protein. Protein made up of 20 amino acids generally, right? These amino acids are created by 3 base pair triplets encoded in the DNA. So, Herbert, if you were looking to find pathways important in the genome, you would look at the DNA. So, what you can do is if you're looking to see how something gets modulated, so you say, like, I'm just going to remove this gene from the genome of this organism, and I want to see what happens. What you can do then is you can look at the levels of RNA and how those change from an organism that has a gene and an organism that doesn't have a gene, right? Exactly, Daniel. Those triplets are codons. Codons. I don't know why they use all these weird language. Don't ask me. I don't even know what a codon is. Like, what is a codon? Who made up that word? Don't ask me. Don't ask me how this all works. I'm just here drinking a beer, hanging out, right? So now we're at the protein being made. So you have the ribosome, this giant thing, right? A ribosome is composed of proteins and RNA, right? So ribosome is not just proteins. They call it an RNP, a ribonucleic protein. Because the cool thing about RNAs is not only can they translate proteins, but RNAs can actually do functional stuff, right? So like tRNAs, there are catalytic RNAs, right? RNAs that can function as enzymes. Their RNAs are kind of a little cooler than DNA generally. But in the case of proteins, RNAs are bound by the ribosome, this messenger RNA, they call it, that turns into protein is bound by the ribosome. And the ribosome then uses tRNAs to match the 3 base pairs codons. And these 3 base pair codons then give an amino acid. And it just keeps doing that. It just goes along the RNA and says, what base pair gives me these, what 3 base pairs gives me what amino acid? Boom. And they just keep adding on the chain, keep adding on the chain, keep adding on the chain until it gets to the end of the RNA and the RNA tells it to stop because there's also these stop codons, right? Like Craig was saying UGA is a stop codon. I'm pretty sure, right? Um, if you want to be really badass, you memorize all the codons and which amino acids they code for. I'm not that good. I only know some of them, but that's how you'd be really cool. How long are RNAs? So RNAs are as long as the DNA encoding for the protein, right? Now, sometimes RNAs can have other elements in them called introns. Introns are regions that hold other information but are generally spliced out before the protein is made. An exon, EXON, is what is generally a protein coding region of a gene, right? Intron, non protein coding region, exon, protein coding region. I know all the terminology is super annoying and complicated. Please don't blame me. Super annoying, let me tell you. So the RNA inside this ribosome, and the ribosome is just adding amino acids from tRNAs, adding amino acids until you get a protein chain. David, so here's the interesting thing, right? You need proteins. You can't just start with DNA. That's the crazy thing, right? That's why viruses aren't alive. So viruses don't have the protein machinery generally to replicate their genome and do all this other stuff. So viruses infect cells, co-opt the machinery already inside cells, the machinery that makes cells alive, and it uses it in order to propagate. So like if you just took DNA and like we talked with Kate Adamala, if you just took DNA and shoved it inside something, nothing would happen. You need proteins already there to do something, right? So proteins essentially create the RNA, they create DNA, and they create proteins. So proteins create it all, but they also create themselves. Herbert, kind of. So generally, when you use resources to look up proteins, so a good resource to find proteins is NCBI database. You can go to the NCBI database, ncbi.nlm.nih.gov. Let me get the link. And you can find just the protein coding regions of genes. You can find whole genes, you can find whatever you want. It's such a great resource that you could find. If you want to look at protein structures in particular, which we will when we have our next class, this is what we're going to do. We're going to learn how to look at protein structures. You can go to the RCSB and you can actually find proteins, what they look like on a three-dimensional level, and you know understand what's going on with proteins. So protein structure, the way proteins fold is complicated, really complicated. In fact, we don't completely understand how proteins fold. We have an idea, a general idea. We can apply, Newton's laws and all these physical chemistry effects, but generally, we can't predict well how proteins fold. We're okay at it. And maybe we recently heard about, Google's DeepMind AlphaFold and how it was able to predict different proteins and different protein structures and things like that. We have a pretty good idea how things work, but generally, we are not good at predicting how proteins fold. Why is that? Well, you have to think about, okay, so you have a protein that's composed, let's say a 20 amino acid protein. A 20 amino acid protein is composed of, let's say, 20 different amino acids. Now, these amino acids are interacting with their solution. So let's say they're in water, they're interacting with each other, and they have multiple degrees of freedom, right? So they can twist multiple ways at each joint in between each amino acid. So now you can imagine trying to solve in 3D space how these 20 amino acids are going to fold up. It becomes a ridiculously hard computational problem. Really hard computational problem, right? Yeah, so Daniel, there was a game, it was called Fold It, where they were trying to use it to like help understand and figure out how proteins fold, by using people to try to you know, move and shake and jiggle proteins and things like that. Um, but generally, these things are much more complex than that. Much, much, much more complex. Generally, it's not something that just an individual can sit down and solve. There are some cases where people can. But for instance, if you can, go to the RCSB website with me and let's type in the ribosome. And let's see if we could find a whole ribosomal structure. Maybe we need like the 70S ribosome. 70S ribosome. Yeah, so type in 3BBX. 3BBX into the search parameters at the RCSB. And once you pull up 3BBX, you can, on the left-hand side, you can click on the structure and look at the three-dimensional structure. So now this is a three-dimensional structure of what a part of the ribosome looks like. So ribosome is composed of RNA and protein together, a bunch of different ones combined in a big mesh, right? Now that shit is complicated, right? So complicated. Imagine trying to solve an interaction problem that's that complicated. It would be virtually impossible. Yeah, that's true. Let me just give a link. So here's a link. Just click on this. And you could see this 3D structure and look at it, right? So in the syllabus, there's a program called VMD that I requested you all download. If you haven't downloaded yet, when you get a chance, download the VMD program. VMD program, what it does is it allows you to look at these three-dimensional structures and analyze them. So here at the RCSB, you can look at them on the website, but you can't do it, as complicated look at an analysis. But you can get a good idea of how complicated these things are. You can see all the different parts of it. And if you want, what you can do is you can change the style from cartoon. Let's go to, you can change it from cartoon to space fill. Now, if you change it to space fill on style, space fill will show all the individual atoms of the protein and the RNAs. And then you could see really how complicated, some of these protein complexes and protein structures can be, right? So style on the right-hand side, there should be a style and then change it to space fill. And then you could see all the individual atoms. and what they look like in this. Looks pretty crazy, huh? All right, have people figured that out and seen it? So, what happens with proteins is that after the ribosome makes these amino acids and links the amino acids together, what happens is these amino acids, in a good number of cases, will spontaneously fold into the protein. So, because of the forces of water, hydrophobic effect, electrostatics, van der Waals interactions, all these things will lead to the protein to fold spontaneously into a three-dimensional structure, right? And this three-dimensional structure is what actually does things. That's where proteins actually start to become functional. They're this three-dimensional structure that's basically like a little nano machine. So instead of, say, electricity controlling a protein, you have electrostatics. So you have tiny amounts of positive and negative charges that can affect their structure, right? You have things like the hydrophobic effect, which is water's interaction with protein. So you can imagine something that's on that small of a scale, the way individual water molecules interact with it affects how it moves and what it's about, right? Its structure and how it moves. Proteins are not a static thing, right? They're not this like static little machine inside a cell. They're constantly moving and jiggling because they're interacting with itself, they're interacting with water, right? Water is constantly moving because of heat, right? When you think about the heat interaction, uh, kinetics, and thermodynamics, water is constantly moving inside the cell, and so are the proteins. The proteins are constantly moving inside the cell. Every single molecule in the protein is constantly moving. And this dynamic nature of the protein lends itself for it to be able to function, to be able to work like a machine. It's not really Brownian motion, Jewel. Um, it's it's actually more complex interactions than that. But Brownian motion of water, I don't. Brownian motion, I don't know if that is Brownian motion of the protein itself will move it around inside the cell. Um, but the interactions individually of the individual atoms, there's a lot more physical chemistry that's going on, that causes these things. So, like, you can imagine regions of proteins can be a lot more static, like structured, right? For instance, if you have a helix part of a protein, this helical part of the protein is it's pretty structured. It's a little bit flexible, but generally it's going to be pretty structured, just like, a beer bottle, right? If you have a beer bottle, it can move like side to side and around, but generally it's going to be pretty structured. But then if you have like an unstructured loop region of the protein, right? You can imagine unstructured loop region, it can do all sorts of bendy and floppy and flippy things. It can interact with itself, it can interact with water molecules, right? Depending on what's going on, what it's near, what the temperature is, all these different things are going to affect. Um, uh, do does the thermal environment cause conformational changes as a function of time? So, the way proteins work is thermodynamically. That's how we try to think about proteins and understand them is thermodynamically. Now, thermodynamics um, are kind of, uh, I don't want to say time independent, um, but they are time independent, right? So the thermal environment can cause proteins to unfold, and the number of unfolded proteins in a ensemble. So generally, when we're thinking about molecules like proteins, um, what we are thinking about is we are thinking about an ensemble of proteins, a bunch of proteins at one time. That's because physical chemistry is more a study of probabilities than it is a study of individual molecules, right? And understanding how individual molecules are affected by things is a, a lot more complicated and doesn't necessarily give us the kind of information that we're interested in. So, what we are interested in is how proteins work on a general level. And for that, we need to look at groups of proteins at once, right? Because it's more like if I subject a protein, let's say, to 37 degrees Celsius, some of those proteins might unfold completely, right, and have no structure, but there's going to be a group of proteins that don't unfold completely, right? And this is based on the thermodynamic distribution of how these proteins are interacting with their environment and all the physical chemistry properties that are involved there, right? Over time, uh, more proteins will probably unfold as the proteins that haven't unfolded reach that unfolded state on the thermodynamic uh, distribution, but uh, it's not necessarily time dependent. It is, but it isn't. I know it's a weird thing to think about, and physical chemistry is uh, ensemble nature of things is kind of difficult to grasp when you want to understand things on a singular level, but it's all we kind of have to work with. Um, Understanding things on an individual level when you get down to this scale is really hard because our systems are not set up. Our mathematical systems are not set up for it. Our physical systems are not set up for it. So it's a complicated thing. Um, our systems are set up to understand things on a macro scale almost. Um, and so it's difficult. There's single molecule studies, don't get me wrong. There are single molecule studies of different proteins and atoms and things like this. And if you take like a single protein and you measure its folding and unfolding like a million times, you can extrapolate that to like what would happen to a protein, right? Based on statistics and the ensemble nature of things. But generally, what happens to a single protein doesn't give you a lot of information. I know that's just a weird thing, but yeah. If you put cells in an electrostatic field, do different proteins get generated? No, it's so proteins are always going to be defined by the amino acids. So maybe do different protein structures get generated, Shiv? I don't think so. I don't think, and by electrostatic field, um, maybe you mean like a specific charge, like a highly positive or highly negative field. Uh, in most cases, this macro scale effects probably won't affect proteins. There are proteins that do respond to electric charge, are electrically gated. These are usually membrane proteins, and they have parts inside and outside the membrane, and this allows for a differential in electric charge, right? this differential in electric charge is basically what can cause things to interact with each other or change or move or modify things. All right. So this week we kind of have a good idea what proteins are, what's going on with proteins, and how they form, and all these things. So, what we're going to do, David, so a protein's function is defined by its geometry and folding, but it's also defined by the chemical nature of the amino acid side chains. So I would recommend that everybody go look up the amino acid side chains, and it's probably in this chapter, and understand. So amino acid side chains differ greatly. There are amino acid side chains that are positively charged, that are negatively charged, that don't have any charge, that are hydrophobic, that are hydrophilic, that have all these different properties. So the geometry and folding of the protein is important, but also the chemistry of that in there. Um, that gives rise to the protein function. So it's a combination of both, right? You can think of a protein as molecular, like a, a molecular factory, a molecular machine. So if you want to, like, say, cut, and as an enzyme, you want to cut a molecule. Say you want to break down plastic, so you want to break down polystyrene. Polystyrene is a specific molecular structure. So the protein has to have the shape and geometry to fit part of the polystyrene molecule in it. But then it also has to have the chemistry to cause the chemical reaction that will break the bonds between the other styrene molecules. So it's a combination of both that allow the enzymes to function, right? So for now, we're going to end right here, right now. And we're going to do another class next week because I think we got a lot of stuff to talk about with proteins. So Matthew, it's proteins. Proteins can basically do whatever you want. So adding different bases, adding different amino acids doesn't necessarily make proteins with new functions. That's the interesting thing, is we can basically do whatever we want with the current proteins we have. So it's not like adding more amino acids or a bigger genome is going to let us do really anything different. But yeah, you want people to study proteins, study the amino acids, memorize your amino acids, right? We'll have a test. I'm just joking. We won't have a test. Um, just use it to impress your friends. You know, it's always fun to recite all the amino acids to somebody. And next week, we will go more in depth on protein function. So, what we can do next week, Esther will talk a little bit about the labs. Hopefully, people will have received their packages by next week. And I want to talk about one of my favorite papers, which is the dynamic personalities of proteins. And is probably one of the most heavy papers you're going to read. Um, and it's probably going to be super, super heavy and crazy. The dynamic personalities of proteins. But I want you, if you can, to try to struggle through it and read through this paper and at least get an idea of what's going on. And next week we'll discuss this because it's really important. It's really important to understand how proteins function, because if you want to genetically engineer something, you really have to understand what those proteins are doing in the organism to genetically modify it, right? If you want to create a plastic eating bacteria or fungus, you have to understand how to engineer a protein to break down that plastic, right? So it's just as important in genetic engineering to understand proteins, biochemistry, things like that, as it is to understand how DNA functions. So dynamic personalities of proteins. I want you all to try to read that paper by next week. Look at the amino acids so you have an idea of what the amino acids do. We'll go over that paper next week. And yeah, I know this is a lot of information, but bear with me. We're just trying to get you familiar with this stuff so that you can have some sort of intuition, be conversant, and be a little knowledgeable on the subject. You don't have to be a genius, and you're not going to learn all this stuff in the time of for the class, right? 4 months is not um, enough time. That's the paper. But it's good to just be conversant and knowledgeable in these things, even if you don't completely get it. So don't feel like that if you don't get it, you're not there, you're losing out. Like this is a lot to learn in such a short period of time. It's a real lot to learn, right? I studied these things for, you know, 8 years, even more when I was at NASA, right? So we're talking like 10 years I studied these things and I still don't understand them. So maybe that's just me, but give it some time. But yeah, read the paper. We'll go over it next week. Next week, Wednesday. And if you have any questions, ask them in the Facebook group. Hit us up. We're always around to answer your questions. But yeah, next week we'll discuss the paper. Until then, I appreciate everybody coming on and listening to me ramble. And have a good evening.